Right. Good afternoon, everyone. I see some of you were here from earlier shows, earlier webinars. So let me just briefly do an overview for people who may be new here. First thing I wanted to say is this event is being recorded. It will be on YouTube, and everything that you see on the screen is recorded, including the chat window, just so we're all clear on that. We are going to be running for an hour again. We have two presenters, a series of questions. Michael New is going to set context again for us, and then in the same way that we have done for previous webinars, we encourage all of the attendees to ask questions, make points, and that sort of thing as we go along, and I will try to pick them up, and certainly after half an hour, 40 minutes, depending on how we go, we will throw it open for people to ask questions. And if anyone has any technical issues as they're going along, just pop it into the chat window and we will try and deal with those as well. So, having said that, Michael, advanced life, do you want to set a context?
Sure. Thank you, Andy. So I'm Michael New. I'm the astrobiology discipline scientist at NASA headquarters. Many of you know me. Some of you don't. Hello. What we're trying to do here is start a conversation within our community and hopefully beyond the standard community of astrobiology about our roadmap. Somebody's getting an echo. I don't know how to fix that. I only have one mic, and that's on my ear right now.
We're not hearing it, so we're not suffering. Just carry on. Right.
Okay, I'm sorry. So the Astrobiology Roadmap is a document that the community produces every decade or so, and it informs NASA about what are the open, compelling questions in astrobiology, and therefore informs what NASA will invest in. It also informs, hopefully, the community about what the research they want to do and what they want to apply to NASA to fund. So that's kind of where we're at for that. Every 5 years or so, we take a look at the roadmap and make little tweaks and adjustments, but the big structure of the roadmap, the overarching goals and objectives, they don't change or shouldn't change except every 10 years or so. This roadmap, NASA is looking for something that is fairly aspirational and inspirational. We're looking for something that really throws a lawn dart way out there in the future and gives us something to work for. So a document that lays out objectives that can be accomplished would take more than 10 years to accomplish is preferable to a document that would lay out goals that we can accomplish relatively easily. Evolution of advanced life is the catch-all phrase that NASA uses to describe two different topics. The first is the evolution of multicellularity. So once you have life and you have unicellular life, how do you go from a single cell living alone to an organism that's got multiple cells that form tissues and develop? The second area we tend to put into this category is macroevolutionary processes, and especially the processes of how of things that cause and how ecosystems respond to mass extinction events, like the KT bolide impact or whatever happened at the Permo-Triassic boundary. And all of this research is focused on trying to understand the conditions that lead to, aid, or retard the development of complex life on Earth, so that when we go looking out in the solar system and beyond, we have some sense of what we're looking for. What are the odds of finding complex life? So that is that sort of thing. The purpose of this meeting is to kick this thing off for this particular theme. There are 5 themes we're investigating. And over the course of hopefully at least this week, we'll have a series of online discussions prompted by the discussion we're having today that you'll all take part in, I hope. And from those discussions are going to influence the roadmap in 2 ways. First, the ideas you guys come up with, the open, compelling questions, are going to influence, hopefully heavily, the people who actually write the roadmap. More directly, the people who write the roadmap will be chosen partially from active contributors to the discussions, because clearly people who are active in the discussions have something to say, and hopefully it's a good thing to say, and so we'll invite them to help us write the roadmap. So that's pretty much all I have. If you have any questions about anything, please ask them now. I'll be here all day, but this is intended to be a community event, so I hopefully won't talk very much.
Brilliant.
Michael, may I ask
you, Michael.
a question, please?
Yes, Frank.
For those people who wish to join, or people who are not here for this kickoff, could you forward to us instructions so that we could invite people that we think should be part of this conversation?
Sure. Andy, are you going to put that information up at the end?
In order to be able, sorry, just to be clear, Frank, you're asking for people to take part in the discussions in the website?
That's correct, yes. There may be people that have not been contacted who should be.
Absolutely. Okay, so, well, the thing, it's almost like he can read my mind. If you have a look in the chat window there, you'll see that Mike has just put up the address of the website. I don't need Siri. I just need Mike. And it's very simple. Go there. It's open registration. Anyone can sign up. And then there are a series of discussion fora where we'll be picking up these questions. And the other thing is, Mike tells me probably by tomorrow, West Coast time, the video of both this session and the previous one will have been crunched and be on the website. So if people want to actually listen and watch what was said, then they can do that as well. So should be open to all. So, Frank, just taking that as my lead-in, let's do some introductions. Who are you? Where are you from? Why are you interested in this?
Okay, so my name is Frank Rosenzweig, and I'm a professor at the University of Montana. And I've been a NASA astrobiology researcher for a number of years, interested in this particular topic as well as mechanisms of speciation. And in the current iteration of our astrobiology work, we have two parallel projects, one in E. coli looking at evolution of metabolic cooperation in simple systems, and in using Chlamydomonas reinhardtii, looking at experimental methods for attempting the independent evolution of multicellularity, a trait that has evolved multiple times in the volvocine algae of which clammy is a member. So I've had long-standing interest in this, really from graduate school onwards, and I'm grateful for the opportunity through NASA to participate in this program. Is somebody coming on after me, or do we?
Andy, I think you're muted.
Sorry. Okay. When my lips move but you can't hear anything, I've hit the mute button. Matt.
Yeah, I'll take it from there. I wasn't sure what was happening. I'm a professor of geology at Juniata College in Pennsylvania, and I'm a paleontologist here. My main interest is in macroevolution and specifically the biotic effects of climate change, how that affects things on a macroecological and macroevolutionary scale. My recent research is supported by NASA Astrobiology, which is how I got involved in this, and that is on the latitudinal diversity gradient over time.
Okay, thank you. All right, what happened prior to this webinar was that Frank and Matt and I got together, and I'm going to skip over this slide for a moment, and we talked about what might be interesting questions to help frame our discussion. And the instructions were very much that these questions were supposed to illuminate but not constrain in any way the discussions. So what we thought would be interesting now is to start moving through some of these questions and invite audience thoughts as we go along, and then towards the end we'll pick it up and give the participants a chance to ask questions directly. So if we have a look at the first question, evolutionary transitions. Frank, I know that you'd written this one down as part of the session that you and Matt and I were in. Can I start with you? And this is obviously in the context of the roadmap. And what is it that we should be thinking about over the next 10 years, perhaps from this perspective to start with?
So when I wrote the questions, or in this particular question, my thoughts were being guided by a book that came out a number of years ago, which was actually called "The Major Transitions in Evolution," and it was a piece by John Maynard Smith and A.R. Zothmary. And they identified a number of major evolutionary transitions, some of which may fall more under the purview of the prebiotic folks. So, for example, they identified the one transition being that from populations of molecules, or from replicating molecules to populations of molecules in compartments, the evolution of chromosomes, the evolution of DNA as genes. What I was thinking about was that those things fell to the other group, and what evolutionary transitions I had envisioned, and certainly the group is unconstrained, I guess, in its what it can throw out there. I was thinking about those transitions like that from prokaryote to eukaryote, and implicit in that the acquisition of organelles, which almost certainly were independent cellular entities, and resolving the conflicts that arose in the formation of organelles and the formation of what we recognize as eukaryotes. I was also thinking about transitions from asexual clones to sexual populations, and the evolution of sex, not just in terms of lateral gene transfer, which is unidirectional in the prokaryotic world, but bidirectional as it is amongst most eukaryotes. Likewise, I was thinking of a third transition, namely that from unicellular organisms, most likely protists of some variety way back when, to multicellular organisms. But I was also thinking about higher order evolutionary transitions, which actually could have important implications for the evolution of complex life elsewhere in the universe, namely the transition that has occurred from that of solitary multicellular individuals to that of colonies. In other words, the evolution of eusociality actually falls within these ETs that I was thinking about. So, for example, eusociality, where you have the formation of castes, where certain individuals forego reproduction in the same way that somatic cells forego reproduction and entrust that to the germline. And then, of course, other transitions that Zothmary and Maynard Smith identified were those in primate societies, which evolved elaborate communication systems, languages, which enabled memes. So I was, in summary, then sort of leaving the the prebiotic, the molecular evolution of replicating molecules and the formation of compartments, evolution of chromosomes, to our colleagues concerned with prebiotic evolution and the evolutionary transitions that I had envisioned for this group were those that are basically to eukaryotes, evolution of sex, evolution of multicellularity, evolution of eusociality, and evolution of language.
Matt, really tack on to the end of that. The first thing I think is that, so far as I know, there's not a group of major evolutionary transitions that have kind of been defined across the field. I think if people put together their own lists, they would have some in common, but they would also have a lot of these events that were not in common. And so I think that as a field, we don't necessarily even have a common understanding of what the major transitions are, or really even what major means in that respect. There are things that I think are not as important in terms. If I was making a list of the major events, I'd put the same things on there that Frank put. The evolution of multicellularity. I'd probably also put things like evolution of bilateral symmetry, or a head. Having a head, something like that. Further back in time, the evolution of respiration. Of oxygen breathers, and things like that. But I'd also probably put things that are less maybe monumental in terms of the overall history of life on Earth, but which are of kind of particular importance to humans. Things like evolving an internal skeleton, vertebrates, evolving things like eventually the amniotic egg, then eventually live birth. And then I might also add in things like mass extinctions, which in the grand scheme of things really just reshuffle the kinds of complex life that are around, and it changes what's dominant at times without really creating any new kind of life in the sense that moving from unicellular to multicellular did, but which nonetheless things like the KT radically altered what kinds of life are around, and maybe even those effects are even continuing today. I think some papers recently have come out showing that you can still sort of observe the effects of the KT and things like the PETM in the modern biota. So I'd probably put those in that same category of major evolutionary transitions.
Great.
So you're adding to this basically, to kind of summarize, major metabolic innovations, major architectural innovations, within clades, and then evolution of diverse developmental strategies, that, as well as these challenges and opportunities of a major sort in the paleontological record.
So let me just ask, the fascinating opening, how would one go about researching this? Because I love the thought of spending NASA's money, and let's assume you were given large quantities of it. What would it mean in terms of a research roadmap? What would you be looking to do?
I've always been struck by the fact how much of that research is discovery-based. How much it's really relying on the generation of new data, not simply the analysis of existing data. That's not to say that analysis of existing data is not important. I do that a lot, but I'm just constantly surprised by how much we don't know, and I think more. This isn't really an experimental approach, but more discovery funding is really important, I think, in that.
And Frank, what would you do?
Well, I mean, I'm an obsessive experimentalist, and so in my group, we're trying to replicate using extant forms some of the. Let me just narrowly define and say these Maynard-Smith / Zothmary transitions, like that of evolution of organelles, evolution of multicellularity and cooperating systems. But I would concur with Matt's remarks and add that some of those discoveries, at least with respect to, say, innovations in metabolism and in architecture and developmental program, some of these major innovations are they're essentially they're entombed within the sequence record. That is to say, the tree of life and the code, which basically gives us the tips of those branches today, contains a lot of information that would illuminate these questions about these major anatomical and developmental innovations.
Go on. I want to add one thing. I see there's a comment from Eric Smith about, he points out that the major transitions here are defined or connected to the rise of atmospheric oxygen. I think there's an important point that some evolutionary innovations they're hierarchically structured in the sense that some of them allow new evolutionary innovations, and I think that sort of hierarchy is an important area to explore.
Yes, absolutely.
Can we also look at John Cave's, well, actually, John Cave II, his question, which is about the thermal vents in Europa?
I don't know enough about it specifically to comment, Frank.
And I must join in with you there. And I guess there are a number of people who have joined since your initial introduction, Andy, and so maybe just indicate to them at approximately what time in this discussion people can come online, and though regrettably invisible, they will be certainly audible, if I understand correctly.
Yeah, right. And the answer is probably in about 20 minutes, although it all depends on just how fascinating your conversation is. Okay, let's move on and look at the comparative methods, and we'll park John Cave's question under, we need to do research on it. Matt.
So go to the second set of questions?
Yeah.
Okay. All right. So we kind of touched on this a moment ago. I just noticed Yuri Gorby signing in, whom I have not seen in a long time. So although I don't see you, Yuri, I see your footprint here. So we talked about information about evolutionary transitions that are embedded within the sequence data in the tree of life, and so that is kind of what I was thinking about when I wrote the question, that we could seek or use comparative methods to draw strong inferences about mechanisms driving evolutionary transitions, and to the extent that our molecular clock data can be roughly calibrated to known events in the fossil record, then we can sort of, retrospectively examine those, the succession of those innovations in different groups over time. In terms of experimental methods for the analysis of evolutionary transitions, again, I'm really thinking about taking, say, extant groups that have no history. Say, for example, in the transition from unicellularity to multicellularity, perhaps a number of you in the community read interesting paper out of Mike Travisano's lab at the University of Minnesota, spearheaded by Will Ratcliffe, on the evolution of multicellularity in yeast. And it's a very elegant piece of work which has been criticized in some quarters by using an organism that has perhaps an ancestry of multicellularity and looking at a derived trait. But there are plenty of organisms that are bona fide unicellular organisms on which we could do experimental evolution. So in terms of methodology, that's what I had in mind, but other people may have equally good or better ideas on this point. Matt, how about you?
I think that certainly that's the same understanding I have of experimental methods. The major evolutionary
and comparative. Let me just jump in today about comparative. I really wanted to emphasize, not just because you're a paleontologist, but I wanted to emphasize that the tree of life data, it absolutely has to be mapped onto the paleontological records. So.
The evolutionary transition that's seems most important these days is the one related to anthropogenic climate change, and I've seen a number of interesting studies related to experimental manipulation of environments that mimic potential future conditions under various scenarios of climate change and what happens to those organisms in those circumstances. I think that we have a lot to learn from studies like that. And then, of course, there are, plenty of analog environments in the past that, in which Earth history functioned as this natural laboratory that has manipulated the Earth for us, and then we can go back and look at for example, selectivity of extinctions or preferential origination after an extinction, and see how those environmental events altered the biota under conditions that we expect to be similar today. I think those would be really informative studies to do.
Before we move on, I wanted to pick up Sean's question, which you'll see in the live notes there, and I'm going to use it to sort of bridge the evolutionary transitions and comparative methods. So this is the one about the presence or absence of O2 and the possibilities of advanced life, which obviously, as Sean says, for people interested in exoplanet missions would be very exciting. What are your thoughts about that in terms of the evolution of advanced life?
Matt, why don't you
I'll say that certainly we have life that does not require oxygen, but from my perspective as a multicellular kind of guy, all the interesting stuff is happening among the oxygen breathers. Sorry, Frank. You work on aerobic bacteria, don't you?
I do, but I have worked on anaerobes. I mean, what's interesting is that there is this electronic scepter tower, say in sedimentary environments, and there are obviously oxygen is the king. However, you can get down to pretty good respiratory mechanisms that are based on iron reduction or on alternative electronic receptors that are pretty energy efficient. And while, to my knowledge, and maybe Yuri can chime in on this point when we open it up for discussion, but while clearly there are cyanobacteria that exhibit the hallmark features of multicellular differentiated organisms, to the best of my knowledge, there are no anaerobes that exhibit those traits. So I may just be exhibiting my ignorance of the literature, of the recent literature, but I'd like to hear more on that from the community. And really, are there any intrinsic limitations to the evolution of more quote-unquote complex forms in anoxic environments?
Okay, that's for those of us watching the chat room. You can see that this has produced a little flurry of comments there, which we will come back to. I just wanted to pick up one thought, though, which is that it's one or two people had been asking, these are sort of arbitrary distinctions we're making between prebiotic and advanced life, and ultimately when we get to the road mapping workshop, we'll be free to structure it any way we want. It's just we sort of have to give titles, so please don't feel that the decisions have already been made. Okay, so let's move on to the part which
I want to add, just because I requested a comment from Yuri, and one came very quickly, and he made the important point that there are anaerobic biofilms, and of course biofilms are multicellular structure, although it's, I think, an area for research and open discussion as to the degree of cooperativity and predictability of their assembly. But a clear and important point made.
Great, thank you. All right, single most important research problem. What are your thoughts on where we might focus resources and effort for identifying the transition from simple to complex life forms?
I'll be very brief here, and Matt can chime in. This kind of follows on from the previous discussion, as well as all the chatter here about oxygen. And to me, particularly if you're wanting to pursue an experimental approach to some of these questions, finding the appropriate set of model organisms and the appropriate analog environment for performing these experiments seems to be of paramount importance. And I would say that the various transitions that we have described, that non-exhaustive list, each of those transitions calls perhaps for a different model organism and a different analog environment. Matt?
For me, I think the most important research problem is really just a general one, which is how resilient are Earth's ecosystems to environmental change. What is the rate and the length of time that a perturbation is applied? How does that affect the outcome? What sort of organisms are better able to withstand environmental perturbations of whatever sort? Which ones are most at risk? How do these structure the biosphere? How has it affected ecosystem functioning, including things like nutrient cycling and that in the aftermath of the environmental perturbations? How are different life history strategies helpful or harmful in getting organisms through those events? Anything I think around ecosystem resiliency is in my mind, the most bang for your buck.
Mm-hmm.
I have to admit, just picking up the chat window for a moment, I do like the idea of one attempting to avoid being poisoned by the concept of oxygen as king. It's a sort of oxycentric view of the world. All right. Now, I know one or two people have jumped forward to this point already, but let's have a look at sort of multicellularity and the superorganism and E.O. Wilson and the rest. What are our thoughts around this? Where does it fit within any future roadmap? What should we be thinking about here?
Well, when I put that down last week, I think Matt has, wisely emphasized the sort of abiotic context in which these biological transitions occur, and sort of biotic constraints or opportunity generators for advancements in the complexity of life. The wording of this last point on the PowerPoint slide is sort of explicitly population genetic, and so when I was thinking of units and selection changing, that is to say, I'm thinking about is selection focused, on a molecule than is it focused on a cell, and how are conflicts resolved between entities within the cell, within the various plastids, and then the nuclear genes, and then on up the ladder, even up to say cooperating metabolic systems, like some of the people are talking about in the open chat. Is the unit of selection, an interconnected biofilm, or does the unit of selection remain focused on a particular cell or even a group of cells, within a biofilm? So that's kind of what I was thinking about. And so, Matt, you want to chime in?
Yeah, I agree. I think especially the unit of selection is important in just understanding functionally how things are going to be able to adapt or not to any sort of future change. So I think that's important. I have to confess I don't actually follow the E.O. Wilson superorganism thread that carefully anymore, but certainly the units of selection have been important in paleontology and evolutionary biology for a long time, and that certainly will continue to be. I guess I've always sort of been impressed by how little we still know about really fundamental things about evolution. Like, I don't know how far we've actually come since G. Evelyn Hutchinson asked why there are so many kinds of animals. I'm not sure we actually even still know why there are so many kinds of animals, and that is a larger and an overarching question to the units of selection question. I think is important. Yeah.
Absolutely.
All right. Well, at this point, given the amount of chat in our chat window, obviously our afternoons are a good time for chatting on this. Let me go open to the participants. Now, Uli, I can see that you've had your hand up for a while, and we've also put your question into the live notes. Do you want to expand on that at all? Basically, would you like to talk, or do you just want to hear the response to that? If you want to talk, do *6 to open your mic. Oh, that sounds hopeful.
Hello, can you hear me now?
Yes, we can.
Okay, sorry, because I had to put the phone down. I didn't hear part of your question. So I think part of the reason why my hand was up is because I'm unfamiliar with the system. But I think I still was thinking in terms of evolutionary transitions, that we could see them maybe very generally as a specialization of molecules, that maybe in many respects we start with molecules that are generalists in many different ways, and then they specialize themselves not only as DNA for genome and protein as major catalysts, but also as membrane proteins that may enable multicellularity to get cells clinging together, so that we have specialization of molecules, and that maybe a focus on molecular evolution and less on the phenotype may give us more insight on the major evolutionary transition.
I still tend to think that the unit of selection is the individual, and I know that obviously that's heresy in some quarters, but or traditionalist anyway. But in that sense, I think looking at the individual as the package in which all these molecular things are happening is important. But I agree that thinking of things as sort of increasing hierarchically ordered levels of complexity is useful.
So in that sense, for example, we can evolve macromolecules in the cells in my lab, and we can do experiments on that, on evolution of molecules. And for example, we are starting to form RNP complexes from an evolving RNA molecule, which is probably one type of specialization of forming catalytic RNPs. So what I want to say is that with this molecular focus, we can we can do experiments. We can look at populations. We can get sequence data, and we can get very quantitative data. Which makes lots of things addressable quantitatively. And as an evolutionary transition, if we consider, for example, multicellularity as being the consequence of which molecule is it that holds cells together? It was, as far as I understood, it was one major molecule that was holding cells together that enabled the rise of multicellularity. Is that correct?
Well, I don't know that perhaps in amongst animals, but I think that in other multicellular groups there are a variety of glycoproteins and lipopolysaccharides. I think it's taxon specific, amongst protists and fungi. But there are limited subset of extracellular matrix compounds that act as the glue. So that it's a finite set, but it's not just one. At least that's my understanding of viewing with the view across the eukaryotic, across all three domains.
Right. Can I ask you, does that imply that multicellularity arose independently in different groups?
It certainly did. There are many independent evolutions of multicellularity, even within fairly narrow clades, such as the bulbocene algae. I mean, everybody who's, all of us, at some point, peered at a Volvox cell, when we were probably in grammar school, and within this clade, depending upon whether you're a lumper or a splitter, say, about 20 organisms, you've had multiple evolutions of multicellularity, and so this appears to have happened a number of times, not in the distant past. Sometimes even within the last 50 million years. So yes, there are independent.
For astrobiology. Go ahead.
No, please.
One thing, just thinking about astrobiology here in general, is which ones of these major evolutionary transitions are the ones that are easy to do and have arisen multiple times, and then which ones have been unique and maybe chance events that are not likely to be repeated? I guess I would expect that if we were looking on other planets, that the evolutionary transitions that are repeated events here seem to be like the most likely to be the ones that are repeated elsewhere. And the ones that are chance events here are likely to be the chance events there.
Yeah. So, for example, I think that the transitions that Zothmary and Maynard Smith chose were ones that had clearly happened, not just once but multiple times. And so, it's the eukaryotic cell. There's not a eukaryotic cell. There are many different kinds of eukaryotic cells, and they have acquired their organelles in different ways, independently. Certainly the lineage that's given rise to animals acquired what is now, or the winner, just like they say history is written by the winners, the evolutionary history is written by winners as well. And so we have the winning mitochondrion, but there's no reason to suspect that in the early going that there were not many experiments that gave the bigger bang for the buck that that some alpha proteobacter way back when gave. So these transitions from prokaryote to eukaryote, from asexual to sexual organisms, from protists to multicellular organisms, you can show multiple evolutionary origins for all of these things.
So let me ask the participants now if there are other people who want to talk, as Mike just put in the chat window. At the top of the screen, the right side of the camera, you'll see a little person with a hand held up. You can click on that, and that lets me know, and it sort of pops you in the queue. And just while people are thinking about that, I've got a comment in the chat window here about no, it looks like I've only got initials, CIW. So this idea of taking O2 off the table, does that mean that we define evolutionary complexity in terms of the emergence of metabolic complexity? Are the two the same at that level?
Mm-hmm. I'm getting comments here by, is it Yuri? Yuri, can you actually chime in so that, in fact, he has his hand raised. So rather than me, distilling and possibly misinterpreting his remarks, I'd like to hear from either him or Eric directly on these points.
So let's go with Yuri and then Eric. So Yuri, it's *6 to unmute yourself, I believe.
Actually, can I talk just on this telephone that I'm on? Because I called in on my phone.
Yeah.
Can you hear me?
We hear you.
Perfect. Hi, Frank. How are you?
I'm well. It's good to hear your voice.
Great. Thanks a lot. It's nice to be here. I just was made aware of this webinar from my colleagues at the University of Southern California, and I don't see any of those, I don't think, on the line here. So we were just awarded one of these NAIs, and I've moved up here to Troy at Rensselaer Polytechnic Institute. So I'm up here in upstate New York. I did want to just kind of go back a couple of comments or lines back in the chat room where I put that link to a paper by my friends and colleagues at University of Aarhus. There are anaerobic eukaryotes. I mean, and again, the lineage of the mitochondria is different than the lineage of mitochondria that, for us, for instance, I think we have rickettsia lineage of mitochondria, if that's correct. And I guess I was trying to read quickly through there, but yeah, I think it's either sulfolobus or one of these sulfur organisms were the predecessor for evidently through molecular means, the predecessor of the mitochondria in these benthic worms and other foraminifera, and there's a number of them. And so I don't think that, again, that we should limit our thoughts or our ideas to oxygen-respiring eukaryotes, since we do have examples here already on this planet. I think that it is absolutely possible that you could have divergent life forms, eukaryotic-like life forms with anaerobic mitochondria that operate just fine in the absence of oxygen. So I think that we shouldn't again limit the possibilities just by from our lack of knowledge of the complexity of biology. So I just wanted to make that point. And the concept of multicellularity and biofilms, many of us that study biofilms look at these not as single organisms. In fact, there's only a small percentage of organisms in multicellular or diverse biofilms that can't live by themselves, and so they are actually absolutely dependent on their community members. And so in that sense, it may not be considered a single organism, but certainly a reactive and integrated tissue. And we do know that within those tissues that power energy can be shared now through electrical means or through metabolites that are released and diffused to other cells within that tissue. And so again, I think that this is something to consider when you think about the diversity and the generation of multicellular assemblages, at least, that could be considered that, and by many of us they are.
Excellent.
Eric, do you want to add on that? Do you want to take it in a different direction?
Hi, Andy. Can you hear me?
Yes, we can.
Okay, thank you. If it's okay, I just wanted to respond to thoughts that are generated by your point 4, which would then be slightly off topic relative to the oxygen point.
Go right ahead.
Okay. My thought is that what we care about in complicated life, as in simple life, is how architectures form that can take on and preserve and use patterns. It's good to not prejudice that too much by thinking about a few familiar kinds of organization. So if we ask what structures evolution, at a bare minimum, we have development in the broadest sense, meaning sort of all the constructive activities within ontogeny. We have transmission, we have selection, and we have ecology. And in all of these, there can be hierarchical nested units of organization. It's very good to talk about units of selection, but it's also very good to recognize units of all the others, because these are the things that determine how variation is structured. They determine what the arenas are within which selection takes place. And it strikes me that what we care about is the emergence of complexity in evolutionary dynamics and also in interaction with the boundary constraints. And to use a language that keeps that field open to studying all of the relevant structures seems to be a healthy direction, especially if you're trying to lay out a roadmap. And I guess in some sense, that was also where the oxygen point came from, to recognize that all of these things really are system conversions within which we may recognize a certain kind of innovation as the marker of the event, but often it's not the only determiner of the event, as we see in many, many transitions over and over again.
Thank you. Guys, thoughts on that?
No, I mean, just an excellent point, and certainly, one to be, it would be nice if perhaps Eric could, well, he has written some comments. As the week goes on, will some of the notes be available to those people who log in and discuss? Because that's actually a point that I would like to see discussed further. It was very eloquently put, by Eric just now. Will a transcript of that be available from this point going forward?
Yeah, what will happen is we'll have the video processed by tomorrow. We'll have the chat transcript. And we're going to experiment to see if we could break out any of these points into separate threads. Certainly, I think it's particularly interesting to think about what are the assumptions or even meta-level assumptions that we need to be aware of in developing a roadmap. So over and above what questions might we have, it's also really interesting to think about what assumptions we must try and transcend in the development of the roadmap, and that sounds particularly important. And in fact, Eric, we'll pop the transcript up. You may want to raise that as a completely separate question in a thread, just to make sure that we don't butcher your language unintentionally in sort of breaking out the threads on that. Okay, Sean has also been popping up some questions here. If you have a look in the live notes, the last one down, do we have any clues on what global signatures? And while you're thinking about that, let me just remind people, we've still got a couple of minutes left. If anyone else wants to ask a question, please stick your hand in the air and we'll come to you.
Yeah, this was a question by Sean. Is that correct?
Mm-hmm.
And it was of the 10 questions that we generated the other day. While the second question was not part of it, the first was, in fact, almost exactly question number 7 that didn't make it to the final 4. And Matt, since you're the paleontologist, would you like to take a stab at that? And perhaps Yuri might also. So I guess the question is, do we have any clues on what global signatures we could look for from advanced life in a globally anaerobic surface environment?
From a remote sensing standpoint, I have no idea. It's just not my area of specialization. But I think on a larger scale, looking at life on Earth and seeing under what conditions life looks a certain way or does not, and then finding those analog environments on other planets is actually answering the same question. It doesn't confirm that there's life there. It just means that the possibility for life under these particular circumstances is there. And, maybe it's not as nice as some sort of satellite that can beam a planet and tell you what life is there or not. But I think it does double duty in the sense that it helps you understand the presence of life or not on other planets, and it also helps you understand the life on this planet, which does exist. And so in that sense, it's bang for your buck in that regard too. I also wanted to pick up here on Uli's point that, and sort of also Eric's point, which was excellent. And one thing that he said is that Uli said is there are many possibilities for a major evolutionary step, and he was referring to Frank's comment about multicellularity evolving multiple times. And it strikes me that there are also many physical environmental events in Earth history that were not accompanied by some sort of major evolutionary step. In some ways, defining the major evolutionary transitions is to say that we have recognized that something major happened to life at that time. And then we go looking for the causes of what happened. But there's also these times when major environmental things may have happened, but that they were not reflected then in anything recognizable to us in the history of life. And so I almost think a useful research direction is something which is not biological at all, which is to look at the absence of biological change during times of major environmental shifts. So far as I know, I don't think much of that would happen in the Phanerozoic, but if you go back far enough, there may be times like that. That to me would be just as interesting in understanding why things evolve complexity, by looking at when they don't.
Mm-hmm.
Okay, and with that, ladies and gentlemen, I'm going to wrap this up. The recording will be available tomorrow morning if you want to mention it to your colleagues, and we will send out a newsletter to make sure everyone has that. The questions which Frank has been referring to, the additional questions which they developed, will be available on the website. However, I've just had a message from one of my colleagues saying that apparently there's an ever so slight problem with the way the threading is working on the website. Seemingly a kitten got in and monkeyed around with a ball of wool. We need to straighten that out. So the questions won't be up today, but as soon as we send out the newsletter, we will let you know that it's back and that we want your input on that. So thank you very much. Very interesting discussion. We shall all go off and ponder. Thanks a lot, guys.
Very well. Thank you, Andy.
Cheers.
Thank you.